Good morning, everyone. Welcome to Twist's webinar, Writing the Future of Biologics to Discover and Optimize Antibodies. My name is Tim Drevens, and I'm the event manager for the Americas for Twist Bioscience. I just want to go over a few housekeeping items with y'all. All lines will be muted during the webinar. We'll save all of our questions for after Aaron's presentation. If you do have a question, please make sure you ask it in the Q&A box rather than the chat box. And following the presentation, if you have 1 minute to take a brief survey, we'd greatly appreciate your feedback. Now I'd like to take the opportunity to introduce Aaron Sato, the Chief Scientific Officer for the Biopharma vertical at Twist Bioscience. Prior to Twist, Aaron served as the Chief Scientific Officer at Lake Pharma, leading the California Antibody Center, which discovers novel antibody therapeutics for its clients. He also oversaw all discovery research functions as both the Vice President of Protein Sciences at Surrozen and previously Vice President of Research at Sutro Biopharma. He also held director-level positions at both Oncomed and Dyax Corp. Aaron, the floor is yours.
Great, thanks, Tim. So again, my name is Aaron Sato. I'm the head of the Biopharma vertical at Twist Bioscience. And it's my pleasure today to give you an overview, first of all, of the Biopharma vertical at Twist, and then second, give you an update on our recent efforts to find antibodies to help fight the COVID-19 pandemic. So again, I always say the best companies out there really understand the one thing they're really good at. And so for Twist, that's our ability to print DNA on the silicon chip that's shown on the right. So our base technology allows us to print up to 1 million individual oligos of discrete sequences up to 300 base pairs in length using our silicon platform shown on the right. And again, we use, so it's basically a fantastic system to basically make all kinds of discrete oligo pools. And we use that to make all kinds of different DNA products for Twist. In particular, our highest revenue grossing product is, of course, our ability to make clonal genes. So you can go online, order specific genes using our e-commerce platform, and then within several weeks, you'll get your clonal gene cloned into any vector you wish. We also can use oligo pools for all kinds of other DNA applications. In particular, we have a whole line of NGS kits that we offer. We also can use oligo pools to make high quality DNA libraries, which I'll talk about in a second as it relates to our biopharma vertical. And then finally, we also use this platform to make all kinds of other DNA products that we're selling recently for the COVID-19 pandemic. And in particular, we've just recently put out a whole series of RNA controls to help with PCR-based testing for the virus. So how do we use oligo pools to encode for and make DNA libraries? So if you think about an antibody domain as basically a collection of 3 different loops or 3 different CDR loops, we can basically make a pool of oligos that encodes for diversity in each of those 3 loops. The reason why, as an antibody engineer, this is really game-changing for making an antibody library is that, unlike in the past where people have used typically a degenerate oligo to encode for this diversity, here I can actually make oligo pools of discrete sequences and then PCR them together to create a hypervariable domain where I have diversity in all 3 loops. This is a subtle difference, but it's important because it allows us to potentially, as we build our library, make sure that the diversity in each of those loops exactly match what's seen in the natural human repertoire. Whereas when folks use degenerate oligos to encode for this diversity, they try to mimic it as best they can, but they oftentimes get unnatural combinations of amino acids that doesn't match the natural human repertoire. In addition, because I know exactly what sequences go into each of these different oligo pools, I also can remove sequences up front that might lead to developability risk. So sequences that lead to isomerization sites, cleavage sites, deamidation, or glycosylation sites. And then finally, which is key to one of our libraries focused on a difficult to drug target, GPCRs, I can also encode for specific motifs in my oligo pools as I build our libraries as well. So again, just to reiterate on the advantages of using oligo pools to build libraries. When we build our libraries, unlike, again, as I said in the past, people have used degenerate oligos, we have really precise base by base precision as we create these oligo pools and then we encode them into our libraries. We have strict control over codons that we use. We have fantastic combinatorial diversity by mixing different oligo pools as we build our libraries, and we have really tight control over the amino acid distribution as we build them. Putting in length distribution, particularly for heavy chain CDR3, is really easy because we can just make oligos at different lengths. Whereas in the past, when you use a degenerate oligo, you would have to actually make separate degenerate oligos for every length. And because we're making each sequence explicitly, we also can remove restriction sites and unwanted motifs, and also, as I said before, sequences that might lead to developability risk. And oftentimes we use encode for multiple germlines as we're building our library. And then finally at the end, which is really important, is we actually QC all of our libraries with next generation sequencing to really validate the quality of the library at the end. So on a whole, what is Twist Biopharma focused on? So again, we're a division of Twist Bioscience, and we basically utilize all of the fantastic DNA tools of the company to help others discover and optimize antibodies. So on the antibody front, I'll show you examples in a second. We have a whole series of different scaffold libraries, either Fab, scFv, or VHH single domain libraries that are all in phage display that we can pan and screen against any library and pull out therapeutic hits. We also have a library focused on G protein coupled receptors. One area that I always call out as a fantastic application for synthetic antibody phage display libraries are difficult to drug targets, and certainly G protein coupled receptors are one of them. So, and as you'll see later when I talk about the breadth of all the libraries that we have, you'll see that this is one of our focus areas that we're actually creating specific libraries on different classes of difficult to drug targets. Thirdly, after you have your lead, you often need to optimize it and potentially humanize it. And we actually have a whole platform that we call Twist Antibody Optimization, or TAO, that can help you with this. And I'll walk you through briefly how that works. And then finally, one thing that goes throughout all of our projects that we do is our ability to make large numbers of antibodies. This is an important piece of every project that we do, both for discovery as well as optimization. And we're actually in the near future actually going to be rolling this out as an alpha product for everyone else to benefit from. And so I'll briefly talk about this today, and it'll also going to be a theme of if you ever need any of this for your own work, you can definitely follow up. And there'll be some questions about this later to get you connected on this. So my vision for our discovery libraries is that, again, in the past, a lot of discovery companies or phage display companies would have one single library, and they would use it over and over and over, and they would be limited by the overall breadth of that library and also the overall diversity of that library, which in phage display is typically around 10 billion different antibodies. My solution to this is because Twist can make high quality libraries so quickly on a monthly basis, I can just continue to expand the breadth of the libraries that I have and create what I call a library of libraries, so that one day I may actually have a plate of hundreds of libraries, each one having 10 billion different antibodies in them. So in total, if I had 100 antibodies of 10 to the 10th, I would have roughly 10 to the 12th different libraries in the end. So that gives me greater breadth in the number of antibodies that I can screen against a target by having the ability to make a library of libraries. So when you look at all the libraries that we've been able to create in the last 2 years since the Twist Biopharma vertical started, it's pretty comprehensive. So on the top right, we have our library series that we call our Hyperimmune libraries, which is a fully human Fab library that uses natural human CDR3 diversity. We've actually created an oligo pool of over 2 million heavy chain CDR3s that we encode into this library. And we've also made a common light chain version of the same library that we can use for making bispecifics. On the top left, we have a series of VHH single domain libraries that we've created. Again, we started with a very large database of llama sequences that had discrete antigen specificity. And then we created 4 different libraries with different design schemes, either based on using ratios of amino acids or just shuffling the natural llama CDRs. And then we also even made a couple versions of the library where we put them into a humanized scaffold over a llama scaffold. And then finally, the last one, which is the VHH human shuffle Hyperimmune, is actually a hybrid library where we have a VHH single domain library where we can have a combination of both human as well as llama-based diversity. Keeping on the theme of just general use libraries, we have another library that we call our structural antibody library. Again, this one starts with from a database of all the known antibodies that have ever been crystallized. And we basically made the assumption that if an antibody has been crystallized, it's well behaved and potentially developable as a therapeutic. And so we took all the sequences of all those antibodies and put them into a design to create a fully human phage display library that we can pan and screen against any target. On the difficult to drug side, we focused on, as I said, classes of targets where it's been historically very difficult to find antibodies to that specific class of targets. So as I said, we started out with G protein coupled receptors. I've talked very broadly at multiple conferences about our GPCR 2.0 library, which is a fully human antibody library that uses motifs that bind to GPCRs as a way to direct them to that class of targets. We've now come up with a second GPCR library that basically uses as an input all of the known GPCR antibodies out there and uses the diversities from those antibodies to actually make a second generation library that we call GPCR 3.0. I won't have any data today on these other libraries, but we do have other next generation libraries focused on difficult to drug targets, and certainly ion channels is one area that we're focusing on. So keep your eye out for a new library focused on ion channels in the very near future from Twist Biopharma. And then second, we're also calling out that carbohydrates as antigens are also a really difficult to drug class of targets, and we're creating a library focused on carbohydrates. So in terms of our overall workflow, because we have these libraries on the shelf, they're high quality, they're already ready to go in the freezer, we can very easily pan and screen them, and because I sit inside of a DNA company, we can very quickly reformat them and express them and purify them and take them through a whole series of binding and functional assays. So shown here is just the overall scheme for doing that in terms of doing cell-based pannings for a couple weeks. We follow up and do NGS and Sanger sequencing for a week. And then, as I said, we can very quickly reformat those sequences to full IgG rather than fooling around with the fragments, and make those high throughput IgGs using the service that I'll talk about in a second to give us antibodies that we can do full cell binding assays as well as ELISA-based assays, and then follow up with functional assays. And we can do all of that within a couple months. As I said, the high throughput IgG service has been something that we have created internally to help fuel and power all of our discovery and optimization programs. But certainly as we move ahead in the biopharma vertical, I've been trying to see if we could offer this up to other customers that Twist already has that are already synthesizing antibody genes with us. And so the idea is that someday you'll be able to come to Twist and not only order the DNA that encodes for your antibody, but also order small amounts of that antibody as well. And so we're kicking off a new alpha program within Twist to basically enable others to do either 1 mil or 8 mil transient transfections in HEC-293 to enable them to make either hundreds of micrograms or maybe even upward of a milligram of antibody for their high throughput antibody applications. And shown here on this slide is just all the different options that we're trying to offer in terms of different isotypes of antibodies. We're also enabling folks to do VHH-FCs, since again, as I showed before, VHHs are one of the libraries that we focus on. We have different buffers that we can use for the elution and neutralization, and then also there's other QC metrics that will apply to the purification and purity of those antibodies. And then the idea at the end of the day is to deliver not only the antibody for your applications, but also the DNAs and glycerol stocks that encode for those antibodies. Okay, so now I'll continue on and again talk to you more about some of the results from some of the library of libraries that I talked about before. So for our GPCR library, the example I've talked a lot about at conferences is our work with GLP-1R, which again is using the GPCR 2.0 library, which is the one that has the GPCR binding motifs in heavy chain CDR3. From that library, we were actually able to find a whole panel of antibodies that binds to GLP-1 receptor, which is a GPCR that's involved in diabetes. And again, its ligand is GLP-1 peptide, and upon binding to GLP-1 peptide, the receptor actually signals the secretion of insulin, which is again involved in lowering glucose. So what we tried to show in this study is, first of all, the antibody that we found, of course, bound to GLP-1R on overexpressed CHO cells. But when we dug in a little bit further, we found that the antibodies also were antagonists of the receptor, which shown on the left, you can see that the antibodies at a high concentration actually shifted the activation curve in this cyclic AMP assay with GLP-1 peptide and actually inhibited its signaling. We went on further to show that when we did a dose titration of this antibody, we also saw nearly a complete inhibition of signaling at a high concentration with an IC50 of around 15 nanomolar. And then we eventually scaled up this antibody, did a full PK study, showed it had a long half-life, which I always say PK is always a great indicator for developability. And so we saw a long half-life from our GPCR motif antibody directed against this target. And then finally, we did a in vivo efficacy study in this model where we're trying to see whether we could actually block the activity of GLP-1 peptides. So this is a glucose tolerance study where we injected the antibody and then several hours later injected glucose. And what we're trying to see is in this study, can the antibody actually maintain a high level of glucose in the mice and prevent the binding of GLP-1 peptide, which again would lower glucose. And that's exactly what we see. So we see great activity both in vitro with this antibody as well as in vivo, again validating this approach that a motif-directed antibody can be used to bind and not only and also inhibit a GPCR target. As I said before, we've created a second generation GPCR library, which we call GPCR 3.0. As I said, and I've talked about this at conference as well, but this is a library where we've taken all known GPCR antibodies from the patent literature as well as marketed and approved GPCR antibodies. And we've used that as an input to create a antibody-focused GPCR library. And so shown here is just one example of an antibody that came out of that library panning that we panned against CXCR5. And you can see that the antibody on the left shows really high affinity binding to CXCR5 transfected cells, as well as in a cyclic AMP assay at high concentration, we see a shifting to the right, shown in orange, where we see, again, an inhibition of activity in this cyclic AMP assay. And we're following up on this lead as a potential antagonist of the CXCR5 pathway. And then finally, as another indicator of our Hyperimmune library, which I said before is our fully human Fab library that has over 2.5 million heavy chain CDR3s as part of it. We oftentimes pan our new libraries against CD3 and try to get a human cyno cross-reactive clone. And as you probably know, getting a human cyno cross-reactive clone against CD3 is actually a really tough thing to do with a phage library. And shown here is just an example of two antibodies on the left as well as on the right, where we see very good binding to human and cyno CD3. And again, we're affinity maturing the antibody on the left to make it even more potent against human and cyno CD3. And shown in the middle is actually the positive control antibody that most people use for human cyno cross-reactivity, which is SP34. And then finally, I'll transition into our optimization platform. So again, which we call TAO, which stands for Twist Antibody Optimization. Here, the idea is we've actually created a software that has a very large human NGS database as part of it. And what the software does is we can put any antibody sequence, whether it's murine all the way to fully human. And what the software does is it looks at your antibody sequence and compares that to the human NGS database and looks for CDR sequences that are similar with a defined mutational space that you set. So for example, I can tell the software, please give me all CDR sequences that are within 3 amino acids of my parent sequence. And once it gives me all those sequences, I can again synthesize oligo pools and create a library focused on that specific lead. And if the antibody is actually a non-human antibody, say from a mouse, I also can humanize the antibody during that process and replace all the mouse-derived CDRs with human equivalents that are similar within a defined mutational space. And this slide just kind of breaks down those various steps again in terms of the software analysis to figure out what CDRs to make, the ordering of the oligo pools to make the library. Again, we put everything into phage display and pan it and screen it against the original target. And the overall process for that is basically, if condensed, is about an 8-week period with a 2-week library build cycle and a 6-week discovery cycle. And so shown at the bottom is just an example where we took a PD-1 antibody and increase its affinity by 72-fold. So just to conclude the first part of my talk, which again is focused on the biopharma vertical, there are several ways you can work with us. First of all the libraries I talked about, we're very open to licensing out those libraries on a subscription basis. We're also actively doing partnerships with pharma and biotech companies around these libraries. We have a whole team in a lab that's operational as we speak, that's really focused on helping those companies discover the next generation of antibody drugs. We also can help you with your optimization work to either humanize and or affinity mature your specific antibody leads. As part of our validation of our libraries, we're doing a lot of proof of concept work, and we probably will generate some leads from that work. Since we're not a drug discovery company, we do plan to partner out those leads. And again, you'll see later with the COVID example, this is a perfect example of how we potentially would partner out those leads as part of that process. Another thing that I kind of skipped over is we do have a whole custom library team that's involved in generating libraries for our customers, and certainly oftentimes we often work with small virtual companies that might design their own library, and they work with the biopharma team to help out with the screening of that, either in phage or in discrete ELISA-based assays. And then finally, as I said, we're rolling out a new product, alpha product, which we call high-throughput IgG. So if there's any interest in accessing this as an alpha, please let us know and we'll follow up with you. Okay, so we'll pause for a polling question, which are, what are the 6 ways to work with Twist Biopharma, and which one interests you the most? Okay, so thanks again for voting. And again, the most popular vote was custom library generation with Twist Biopharma, which is great. Share the results. Okay, cool. All right, excellent. All right, so now I'll transition into the COVID example part of my talk. So again, as many of you know, there's been a flurry of publications out on how the virus SARS-CoV-2 is involved in the disease of COVID-19, and also how the virus enters cells. And again, there's this paper that came out recently, earlier this year, that basically, again, conclusively proved that SARS-CoV-2, unlike its closely related viral predecessors, SARS-CoV, actually uses ACE2 and also a TMPRSS2 as part of its entry mechanism into lung epithelial cells. And so this is a really fantastic paper and really important to show that this is the co-receptor that's involved in this interaction, and it's really important for us to understand how we can potentially block this interaction to neutralize the virus. So just to, again, schematically to show the ways potentially that we could create antibody therapeutics to prevent the infection of the SARS-CoV-2 virus. Of course, binding the virus itself is probably the primary mechanism, as I just showed you with that paper. And as I'll show some figures from it, they definitively showed that the S1 protein is involved in interactions with ACE2 on human target cells. So of course, simplistically, you can think about either blocking one or the other of those 2 receptors. So on the virus side, you potentially could think about binding to the S1 protein, and we'll talk later about how there's a specific region of that protein called the RBD protein, that's RBD site, that's actually really key for binding to ACE2. We also potentially could think about binding the human receptor that's involved in entry, or ACE2. Again, this is has been shown previously with other viral infections like HIV, for example, where folks have created antibodies to co-receptors involved in HIV to block viral entry. So you could think about not only binding to the viral target, but potentially also to the human receptor as well. And again, this paper showed really elegantly that when they aligned the RBD, or the receptor binding domain of S1 that's involved in the interaction with ACE2, they were actually able to show that all the strains of viruses that actually still bound and retained binding to ACE2 had a lot of homology in that specific region and had a lot of similar sequences as well. So again, it makes sense why the virus continues to bind ACE2 and uses that receptor to gain entry into our cells. And there are examples of other viruses that show less similarity in this region that don't use ACE2 as their entry receptor. But for the sake of the SARS-CoV-2 virus, that's our main interaction we're thinking about, is trying to prevent the interaction of the virus with ACE2 and trying to find antibodies that not only bind with high affinity to the virus and the S1 protein, but also prevent its binding to the ACE2 receptor. Another series of papers that, again, has been really helpful to understand how to build and find therapeutics to the SARS-CoV-2 virus is there's been a flurry of structures as well that have been really helpful to understand how to find therapeutics against this class of targets. And so there was a really elegant crystal structure of the structure of the SARS-CoV-2 COVID-19 S protein. And you can actually see, it's actually a trimer where you can actually see how the multiple domains come together to form this trimeric structure, which is on the viral membrane. And there's actually one RBD domain that actually pops out of the top of the trimer that, again, is available for antibodies to potentially bind and neutralize the virus. And there's even been additional cryo-EM structures as well with the overall complex of ACE2 with BOA-AT1, which is an ion channel on the surface, which again is on the surface of our lung epithelial cells, and also in complex with the S1 RBD protein. So you can really see the overall interactions from a structural standpoint of how the viral RBD protein interacts with ACE2. And that gives us a lot of clues in terms of what residues and what epitopes we might want to target on both the virus RBD protein as well as the ACE2 receptor. And again, the authors went on further to show that, again, when you line up all the ACE2 binding viral S1 proteins, you can really see how they're similar, particularly in this RBD region, with a lot of similar residues between the SARS-CoV-2 RBD sequence and some of those closely related viruses. All the residues that are shown in blue down in this panel actually indicate the key residues from the structures that are actually important in that interaction. So again, this is the site that we want to bind to with potentially our antibody therapeutics and inhibit the interaction between SARS-CoV-2 and the ACE2 protein. So, so again, for the Twist Biopharma vertical, again, we're trying to validate our libraries. At the same time, we've also wanted to see whether we could help out in terms of potentially finding antibody therapeutics that could help out with the COVID-19 pandemic. And so in mid-March, we partnered up with Vanderbilt University to actually see if we could actually create a library focused on sequences derived from a COVID-19 survivor. So we partnered with the Crow Lab, who actually had a whole series of sequences derived from a COVID-19 survivor, and we used that as an input to create a synthetic antibody phage display library that potentially would be rich in things that could bind to and inhibit the viral entry of SARS-CoV-2. And so in late March, we received those sequences, and we then went on our way to actually create a library from those sequences. And so shown on the right is just, again, different approaches for potentially tackling this virus. Of course, we've heard a lot about potentially making a vaccine to the virus, and then there's a flurry of companies that are involved in this process. But as we know, it's probably going to be a little bit of time before we actually have a vaccine that we can use. And so in the meantime, coming up with antibody therapeutics that are either derived from folks that have already been infected and cleared the virus and already have immunity. When there is, of course, a classical way of basically isolating their antibodies and using them to potentially treat others to help them clear the virus. We also can think about, as we've done in the past, make an antibody derived from synthetic antibody libraries that may also be able to do the same thing. So again, as I said, we've created this library using sequences from Vanderbilt. We've also will take our own libraries that are on the shelf, like the Hyperimmune library that I talked about before, as well as our single domain libraries, and again, apply this to this particular project to find antibodies that not only bind the S1 protein and inhibit the virus from binding to ACE2, but also from the ACE2 side, can we also find antibodies that bind ACE2 and block the S1 from binding from the virus as well. So this slide goes through the overall scheme of how we are planning to go through the campaign. So again, our two targets are SARS-CoV-2 S1 RBD, as well as the ACE2 receptor ECD. We've taken now these three libraries, our Hyperimmune Phage library, the COVID-19 library that we created in collaboration with Vanderbilt, and also all of our single domain libraries. Each of them have a diversity of greater than 10 billion different antibodies. We panned them for 3 successive rounds against each target, picked 3 by 384 well plates for each of them, and screened them in phage display. In order to rapidly make all the hits derived from these pannings, we actually used linear templates. So while we were making the synthetic genes through our clonal gene manufacturing workflow, we actually PCR'd up all of the antibody genes and made linear templates and transfected those into HEC293 cells, and we use that as a process to quickly make IgGs while we're waiting for those clonal genes. And then finally, we have just received this last week those IgGs from those rapid linear templates, and we've now done some initial testing for affinity, and we plan to do receptor ligand competition assays and developability assessment. And so within a 6-week period from library design, we now have putative leads in hand that we can move forward to that are potentially therapeutics for this space. This slide shows you the overall scheme for the panning that we did. So again, we did successive rounds of panning using bioattenuated antigen on streptavidin beads. We decreased antigen concentration between rounds to drive to higher affinity. And as you can see, we had 4 different panning campaigns with, again, different targets and different libraries, as I showed in the previous slide. And as you can see, we saw nice enrichment of titers as we went to successive rounds, again, indicating that the panning was working in terms of driving potentially to antibodies that bind each of the respective targets. And then shown here on these next 2 slides is just some example phage ELISA data. So again, we're seeing really nice target to background binding for all 4 campaigns that I showed in the previous slide. And again, we then transitioned and moved all of these leads forward into those rapid linear templates, cloned as a full IgG to assess their affinity and also put them through a whole battery of different tests that we're currently doing. On this slide, we actually try to combine all of that data. Another assay that we did is we also wanted to see whether any of the clones in ELISA would compete with their natural ligand counterparts. So in this case, for the SARS-CoV-2 S1 RBD binders, we wanted to see whether they were competed off by ACE2, which again would bind to S1 RBD. And as you can see on this dendrogram that we've created, so in the middle, you can see how the sequences are all related to one another in terms of their primary sequence. And then on the outside in green, we've actually plotted the target to background signal from the ELISA I just showed. And then finally around the ring, we've actually put on the phage ELISA competition data. So all the clones that are in blue, we actually saw a high level of competition with the natural protein ligand for the S1 protein, which again is ACE2, indicating that we potentially may have some clones that potentially could inhibit the interaction between S1 and ACE2. And then similarly for the ACE2 binding clones, we did the same thing. But in this case, we did a competition with full-length S1 to see if we could actually compete off that signal. And now again, we see a whole series of clones where we see high level of competition between S1 and ACE2 from the ACE2 side. So again, it looks like from our initial data, we have a wide panel of hits that bind either the S1 RBD or ACE2 and are also competed by the respective protein as well. We then went on and did further specificity ELISAs on the panel of leads, in particular for the S1 leads. And we wanted to see, because again, we had panned on RBD, we wanted to make sure that those leads also bound to the full-length S1 protein, and we actually indeed show that. We had also made the S1 RBM, which is a smaller piece, a smaller motif within the RBD protein. And what we find is none of our leads bind that specific region. I think it's most likely due to the fact that that smaller region doesn't really fold up well like the RBD does. And so it's not really a good surrogate for that region within the folded protein. And so again, we were really pleased that all of our leads, nearly all the leads that bound the smaller RBD domain actually also retained binding to full-length S1. And then finally, within this last week, we recently also got affinities on a panel, a subset of all those IgGs I just showed. So in general, on the ACE2 side, we're seeing subnanomolar binding. And these are all monovalent affinities to binding to ACE2. So again, we're really pleased to actually see picomolar affinities for binding to the ACE2 receptor. And then on the S1 side, we have seen, again, nanomolar, even one clone subnanomolar that's binding to S1. And again, this is just a subset of all the IgGs, and we plan to get even more data showing that we have high affinity binding to both S1 and ACE2. And we're going to be doing a whole series of competition assays to show how well they compete with this interaction. And any leads that come out of this, we'll also potentially put them through an optimization scheme using tau to optimize them further to potentially give us a whole panel of picomolar antibodies. So just to conclude, within 6 weeks from library design, we have leads in hand. For SARS-CoV-2 S1 protein, we have over 51 leads that we're interrogating now. I showed you a subset of those in the last slide in terms of their affinities. And as you can see, we're seeing nanomolar to subnanomolar binding to the S1 protein. On the ACE2 side, we have over 100 leads that we're interrogating. And again, as you saw, we have a whole series of clones that are even in the picomolar range that may potentially be really, again, potentially potent antagonists of this interaction between S1 and ACE2. And then the future activities we plan to continue on with this affinity assessment, do a full panel of receptor ligand blocking, blockade assays, and a full epitope binning using our Carerra LSA platform. We also plan to think about ways that we can multimerize some of these antibodies, or maybe make some bispecific constructs to make them even more potent in terms of either making, potentially making some biparatopic constructs to increase their overall affinity and potency against the virus. And then we're already set up to do a whole panel of developability assays, either BVP ELISAs, SEC, as well as thermal stability using our UNCLE platform. And then finally, we're setting up relationships now with CROs to look at the in vitro potency and functionality of these antibodies with pseudovirus reporter assays, as well as potentially BSL-3 neutralization assays with lung epithelial organoids. Okay, so we'll jump into the second poll question, which is for our high-throughput IgG service, what additional offerings would interest you? Okay, so the top answer was the ability to express other receptor Fc fusion proteins. Okay, great. And again, we'll end there and be happy to answer any questions you might have.
Awesome. Thank you so much, Aaron. On behalf of all of us, that was a wonderful presentation. Just a reminder to our folks listening in, it's time for the question and answer portion of the webinar. If you do have a question to ask, please make sure to ask it in the Q&A box rather than the chat box. I see a few questions have come in throughout the webinar, so let's begin with for you, Aaron. Is Twist looking to develop these leads on their own, or is Twist looking for partners?
As I mentioned, Twist is not a drug discovery company yet. So we're primarily focused on discovery and optimization. So we're definitely looking for folks that might be willing to take these leads on and develop them further to help others in a very short period of time.
Awesome. Our next question from Kath Hutchings. If you repeat library selection using the same library multiple times, do you just observe different diversities each time, or do you see an overlap?
Great question, Kath. Not for the COVID project because we didn't do it multiple times, but for other projects we have done repeat pannings, and we do see different things coming out. So again, that's just telling us that the libraries that we're using are very diverse, and we're just taking a sampling of the total possibilities as we do our panning. So it's just telling us that our libraries are really rich in potential leads. So yeah, great question, and we have seen different things coming out when we do repeat pannings.
Awesome. We have a question from Lee. What other proteins from COVID virus can be good antigen for screening other than S1?
I kind of skipped over it, but there again, as I said, there's a TMPRSS2 is involved in the cleavage of the S2 protein, which is involved in the activation of the virus to enter the cell. That potentially is another target to potentially inhibit, so you can inhibit the cleavage and entry of the virus. There potentially may be other epitopes on S1 other than RBD that might work in concert with the RBD antibodies to think about. And then, of course, others are looking at other antigens on the virus, not necessarily for inhibiting the entry of the virus, but also just for creating a whole series of different detection assays for the virus. So I know a lot of folks are looking at the nucleoprotein as well as other proteins for developing ELISA-based detection assays.
We have time for probably 2 more questions. So this next one's from Andy. He says, thanks for the great talk. My question is how you design the library diversity, especially in CDR3.
Again, as I showed before, for when we build our libraries, we basically make oligo pools and then put them together to build a library. And so the nice thing about that is that we can use the natural sequences derived from those human sequences that we start with. So when we built, for example, the COVID library, we just took those initial sequences from the sequences we were given and just put them in different, and basically synthesized them and shuffled them in different contexts to give us leads that now can bind and have high affinity binding to the viral coproteins.
Excellent. Our last question for today is from Babu. Any other infectious disease targets you are currently working on?
Not currently. I mean, we're about to publish a paper in the next couple of months showing our ability to make large numbers of antibodies to Ebola. We didn't discover those antibodies, but it's more of a paper that we're trying to put out showing the power of our high-throughput IgG service, where we actually have taken hundreds of sequences that bind to Ebola, and we actually show that that we can use that in concert with our Carerra platform to do a full epitope bin of all those antibodies. So it's a nice really highlights the power of our ability to make hundreds of antibodies as well as to do all the downstream testing with our Carerra system.
Awesome. Well, once again, Aaron, thank you so much for a wonderful webinar. I think the response has been overwhelming, so thank you. Just as a reminder to our viewers, if you do have 1 minute to spare, please just hang on. You'll be rerouted to a survey. We'd greatly appreciate your feedback. For our friends overseas, thank you so much. We hope you enjoyed the rest of your night. And for our friends here in the Americas, have a great day. Have a good one.
Great. Thanks, everybody.